Okay, so like Andy said, I'm filling in for Jeff here, and I probably won't fill the full time frame, but we'll save plenty of time for questions, and I might not fully understand some of Jeff's slides. I put some of this together, but pulled a lot of it from some of Jeff's previous presentations. Oh, thanks. Sorry. And so why should we fractionate soils? Well, it's really this idea that soil organic matter is made up of thousands and thousands of distinct compounds, and we're trying to reduce this variability into things that we could start understanding, whether it's just their chemical composition or the functions they might perform in the soil. So lots of reasons. It's really just to try to get more homogeneous fractions that we might be able to make some comment on. And just a couple examples of just looking at soil organic matter. This is a solid state 13C nuclear magnetic resonance spectra. And it's really just showing that there's usually NMR spectroscopy in like biochemical applications, you have these sharp delineated peaks representing individual compounds, but there's so much overlap, we just get these broad peaks occurring. And so literally there's thousands of individual compounds that are contributing underneath these broad peaks. And then another example, and this is just showing, so this is electrospray, ionization, Fourier transform, ion cyclotron resonance, mass spectroscopy. And it basically what it does, it blasts apart organic matter into its individual elements and it's measuring the mass to charge ratio, and you could plot it in this Van Krevlin space, and you could just see there's literally probably 3,000 or 4,000 individual points on that diagram. And so that's what we're really looking at when we're looking at soil organic matter. So yeah, the question is, can we reduce this variability? And it's a question that's been around for, or at least 30, 40 years. 30, 40 years, a couple hundred years, people have been fractionating soil organic matter. And I guess there's kind of a range of methods that are been applied throughout the years. Physical fractionation, just using size separation, just different sieves, different size material has different properties. You can also use ultrasonic dispersion to break up aggregates. I guess that would fall under physical fractionation. Various density separation methods where we know different clay minerals have different densities, and you have different types of organic matter associated with different types of clay minerals, so you could kind of float off. And also just the stuff that's not associated with any soil minerals could float off at a lighter density, and as you get to heavier and heavier densities, you get different clay organic matter associations settling out. Then there's a whole variety of chemical fractionation methods. I mean, this is the classic humic, fulvic acid differentiations where we use different extractions, different extractants, different pHs. And then also, there's a whole class of fractionation schemes that really rely on this idea of resistance to chemical oxidation to isolate a more labile and a more recalcitrant fraction of organic matter. And then there's been various combinations of these physical density chemical fractionation approaches proposed throughout the years to isolate various more meaningful carbon pools. And it really, I guess the choice depends on what you're most interested in, and I guess a lot of science really comes down to really methods need to fit the purpose of the investigation instead of just going in with a predetermined method in mind. And so dating back to some great work that really originated out of this campus, this idea of there's this aggregate hierarchy model to soil, and that's been applied to soil organic matter, in particular the idea that you have organic matter directly associated with minerals, and then aggregates of clay particles kind of condense around other bits of organic matter, and then you kind of get this hierarchy from individual organic matter, clay interactions to microaggregates to and then these microaggregates are then aggregated within larger aggregates, and versus kind of this idea of this unaggregated material, just this particular organic matter floating around in the soil, which might be more susceptible to microbial processing, where the stuff that's associated, especially inside these microaggregates that are within macroaggregates, are probably much more protected than stuff that's just floating around without any sort of mineral protection. And so this idea, this using this idea of an aggregate hierarchy to fractionate soil organic matter has been since Modes proposed this in the mid-'80s, has gained a lot of traction, and people like Julie Jastrow has really extended this, where she's looked really at a lot to do with the fungal hyphae meshing these aggregates and their contribution to stabilizing these aggregates. And then Johann Six, researcher at UC Davis, University of California, Davis, has really pushed the methodology, and it's pretty successful in isolating and actually physically isolating each of these pools and then be able to measure the characteristics of the organic matter. And I apologize, I forget where I stole this slide actually from. And then I guess what I'll call the Adelaide Rossi approach. This is something that our research group has developed over the last dozen or so years, where kind of consider, and I should say the reason for coming up with this particular fractionation scheme is really to try to see if we could create measurable pools that we could feed into a modeling context. And so basically divide all the organic matter we find in the field into the crop residues that are on the surface of the soil, the buried crop residues, so these are your large roots that are retained on the sieve after sieving the soil, your particulate organic carbon, which is defined as material that gets retained on a 50 micron sieve after sieving the bulk soil to 2 millimeters. And then the humus fraction is simply the stuff that passes through the sieve. But then we also have the resistant organic matter, which is really charcoal, and that charcoal could actually exist in either of the coarse or fine fractions, so the particulate or the humus, and we have to use other methodologies to determine that, as it's not as easy as just using a sieve to pull it out. We wish it was, and I'll talk about that a little bit more. And I guess, this idea that we go from more labile to more resistant as we kind of go down this spectra. We get more energy dense, more nutrient dense, so the C to N ratios, C to P ratios decrease, so there's more nitrogen and phosphorus for every unit of carbon as we go down. And this is stuff that's pretty well established, at this point. And so it's a very simple fractionation scheme, but it has given us a lot of insights, and that's one of the reasons we keep using it to today. And I guess, so just saying that basically what we define as soil organic carbon is the stuff that's less than 2 millimeters, so the particulate, the humus, and basically this resistant pool. And here's some scanning electron micrographs that Jan Skensad took of these isolated fractions, and just give you an idea that they do physically look very different. So the particulate fraction, there's still recognizable bits of the plant tissue structure in there. The humus material is really just these coatings on clay particles, and then the charcoal fraction, I think that was after UV photooxidation treatment. You could really see that it's just this condensed aromatic structure in there, stuff that's not very palatable to microbes. And yes, this picture again. It's this idea that we could generate more homogeneous pools. And just looking at some NMR spectra again, we do see that have reduced the variability quite a good bit. So the particulate fraction, we now see, so if you're not familiar with looking at NMR spectra, basically the peak centered around 80 ppm on the chemical shift scale. That's the O-alkyl carbon, so that's your carbohydrates. So the more dominant that peak, the more it's just simple plant tissues, basically, and highly degradable stuff. So we do see that we have reduced. There's still other stuff around, so is there a pointer? I guess not. So, oh.
Against the wall.
It's actually a car antenna. So the idea, actually here, this is actually, you see this kind of triplet of peaks here. This is also, that's really indicative of lignin as well. So we do say there's actually a lot of lignin in there. So it's not necessarily all very labile material. There's some stuff that the microbes might not be super happy trying to digest if there's more energy-rich, easy to digest things around. And then we see the humus fraction. We still have a lot of variability, and we see that it's really rich in, I guess, just two things to point out. So the kind of alkyl peaks, this is a lot of microbial byproducts sitting here, and then this is actually the carboxyl peak, so it's showing that this is highly reactive stuff that's going to bind strongly to functional groups on surfaces of clay particles, so it makes sense to see a spectra like that. And then, say this big enhancement in this peak here, this is probably due to a lot of charcoal actually sitting around in this humus fraction. And then we see after basically UV photooxidizing this same sample, we see that this one peak now almost completely dominates the spectra, and that's pretty much just charcoal right there. So yeah, this scheme does work. There's still a lot of variability within the particulate and humus fractions, but we are starting to reduce that variability. And let's see what else we have to say. I apologize, I'm not totally familiar with all of these slides, so I'm kind of guessing as we go a little bit. And so I guess it's like, okay, so here's bringing the fractions into a modeling framework. Why do we want to know about soil carbon fractions as opposed to just measuring bulk carbon stocks through time? So here's a typical scenario where we're starting at some higher carbon baseline and we initiate a wheat fallow rotation. Our stocks kind of steadily decline. They're starting to trend towards a new equilibrium with that new management practice. And then say at whatever year this is, about year 35, we convert to permanent pasture. And so now we're returning a lot more carbon to the soil, so we start to build the stocks rapidly initially, and then they start plateauing to new equilibrium with respect to the productivity of that pasture. So yeah, we see that actually it only took 10 years to rebuild what took 18 years to lose. So that's really good. We could gain the carbon back pretty quickly. Yeah, there we go. That's what it said there. And now if we look at the actual fractions, we actually see a slightly different story. So for all intensive purposes, this resistant pool basically isn't responsive on management timescales, so on these decadal timescales. So we essentially just set it as this inert pool. We know that charcoal is not totally inert. There are things that happen to it, especially freshly produced charcoal can degrade a little bit, but in the scheme of looking at decadal changes, it's a pretty decent assumption to say we don't need to worry about it. And so what we do see, the particulate pool, we see basically ran down to almost zero under the wheat fallow system, and then that built up really rapidly, where we see in the humus pool kind of running down steadily through time, but it's only rebuilding slowly. And so we actually have a really different distribution of the fractions, even though we have the same total carbon content. So yeah, so we actually end up at this plus 10-year time frame with basically, I don't know what percent, but 30% less humus than we had 18 years prior, the last time we had the same carbon content, and much more particulate organic carbon. So this is it's a lot of implications for, I guess, the resilience, if you're thinking in terms of carbon credits, what might happen to the soil if we put it back under cultivation, and also in terms of what functions this organic matter might provide, since we have much more of the carbons now in the particulate pool than the humus pool, so it might be much more available, actually might provision more nutrients in a short time frame, but you might be able to, you'll probably run through that nutrient reserve much quicker because the humus pool is kind of the ultimate reserve of this kind of long-term, slowly available nutrients. So I guess the take-home message is just looking at the total carbon really isn't going to give you the whole picture of what's happening in terms of its resilience to change and in terms of what functions this organic matter might be providing to the soil system. And so here's just a few examples of different places we've measured these various carbon pools, and we can see pretty different distributions. Some sites, so Pasture and Hamilton, Victoria, have pretty equal amounts of particulate carbon and humus carbon, where we see a cropped, I think this is a continuous cropping site. In South Australia, there's only about half as much particulate carbon as humus carbon, and there's a really high proportion of resistant carbon at that site as well. So we see these vary significantly across the landscape. Excuse me, and yeah, just getting back to this idea that there is large difference in carbon to nitrogen ratios, there's carbon to nutrient ratios in general of these different fractions. So this is showing basically the range of values for some of these different pools. So we see that the surface residues vary from 20 to 100. So our leguminous crops have pretty low seed-end ratios, so they have a lot of nitrogen in them, where like post-harvest wheat, for instance, will have very little nitrogen left in the stubble. And then the roots typically have a little more nutrients in them. And then we see this range narrows and drops, and it's starting to, by the time we get to the humus pool, it's starting to approach that of what we expect the microbial biomass to be. So in this range of about 6 to 8. Potential mineralization of carbon from soil organic fractions. So I guess this idea that indeed these different fractions have different relative degradabilities, so the decay rates are different. So we see the whole soil, I'm just looking at this plot on the left in red, that, yeah, a little bit gets degraded. So, and we see that the greater than 53 micron fractions, so primarily this particulate stuff, is much more degradable, and the humus on average is slightly less degradable than the bulk, which makes sense as the bulk should be kind of a weighted average of the two, and this soil probably had a lot of humus carbon in it, so it's pretty much the bulk is tracking the change in the humus. And then, I guess on your right, looking at the cumulative CO2 emissions during an incubation before and after treating the humus fraction with hydrofluoric acid, and hydrofluoric acid basically can demineralize, get rid of the minerals, since I can't say that word right now. So basically, if we break that clay organic matter protection, is the stuff more degradable? And we do see that after HF treatment, and generally the HF, this is just the 2% treatment. We don't lose a lot of organic matter during this treatment. It's something we do quite often. We do see that the decay rates increase after we break that organic matter clay bond. So it's showing, I guess, getting at some of these mechanisms of why the humus fraction with these clay interactions is less degradable. And here's a slide of my own work, so I should be able to talk about this one. So some of the work we're doing right now is looking at introduction of permanent pastures into formerly annual grass-dominated systems. And we're really focusing on subtropical perennials that are C4s, and wherever these have been sown into formerly C3 winter active pastures, we could actually use the stable isotope composition of the organic carbon to help discriminate between any of the new carbon coming in from the perennials versus the old carbon that was in there from the C3 grasses. So this idea that, so basically C3 plants have a different discrimination for 13C and 12C, and so you get values of about -27, -28 on this delta 13C scale, where the C4 vegetation is more around -13, -14. And we could just apply, wherever our sample falls, we could just apply a mixing model between the two to say how much of that new carbon is in that soil. And so here's a typical profile of an annual system where we have the bulk, particulate, and humus just fall on top of each other. The values get slightly more positive with depth. But then after, I think this site, I forgot to put it up, I think this is 12 years at Kikuyu in a sandy soil in southern district in Western Australia, we see there's been a large shift towards more positive values, which means it's contribution from the perennials. And the reason I put up this slide with the fractions, we actually see that there's much more incorporation into the particulate pool than the humus pool after 12 years. So this is getting at the mechanisms of how carbon enters and gets retained in the soil. So I guess our conceptual model that plant residues essentially enter the soil, they get broken down into this particulate pool, and that's through continuous microbial processing that slowly gets transformed into the humus pool. That conceptual model seems to fit pretty nice to this data, where most of the increases happen in the particulate, and then it's slowly flowing into the humus pool. So we would have seen, if most of the carbon was just, say, soluble exudates coming out of the roots and binding directly to the clay particles, we would have had more enrichment in the humus pool than the particulate pool. So just another example of how this fractionation scheme can be useful in giving us much more information than just measuring bulk properties of the organic matter. And so how do we do it? So this is the fractionation scheme as we currently run in the lab. So we take our 2 millimeter sieved dried sample and we disperse it to get rid of any small clay aggregates. And then we use actually an automated sieve shaking system now to try to eliminate any sort of user variability in the lab. And so basically it just vibrates and shakes, spritzes a little water on the sample, and we get, after a couple minutes, we get our less than 50 micron fraction going through the sieve, and then anything retained on the sieve is in this coarse this particulate fraction, more or less. And then the next step really is that we know charcoal exists in both of these fractions. There's no easy way to get at it, so we use 13C NMR spectroscopy on both of these fractions now to define how much charcoal is present, and then we just essentially virtually subtract out that number and have our third resistant pool. So that's basically what's being showed here. Like I just said, there's some charcoal present in the particulate fraction, so these are little bits that you could often see, the dark little shiny black bits in the soil. And then quite often most of the charcoal is actually present in this really fine fraction. You don't really notice it as individual char compounds, but in the past we found generally 75% or more of the charcoal exists in the humus fraction. In many samples, 95% or more exists in the humus fraction. You don't really see much in that particulate fraction. So I guess that's one change from the work that's been done in the past. We've only measured charcoal in the humus fraction. It was assumed that there was no charcoal in the particulate fraction, and so this is a change from all the stuff that this group has published up until basically anything coming out of our lab from now on. So starting with the SCARP program is using this approach where we're looking for charcoal in both fractions. So it might change some of the numbers from some of the long-term trials and stuff we've done in the past. And that's something, we are looking back at a lot of them, so some numbers might be changing a little bit, but we are recognizing that we might have been missing a little bit of that charcoal in the past, so hopefully the data is better now. And also, what's changed a little, going to this automated sieve shaker system, we've noticed that the breakdown of the particulate in humus fraction, the relative proportions of the two are actually slightly different. And we think what was happening when we used to manually push the soil through the sieves, you probably broke down some particulate matter just by the force of having to work the soil through the sieve. And so we were pushing too much carbon into the humus fraction. And so now we're actually seeing that there is, in fact, when we're a little more gentle on the soil, there's a little bit more particulate fraction around. Okay, so I kind of hinted at this a little bit earlier, but how we use the NMR can be used to define how much charcoal is present. We know charcoal has this really distinctive peak centered at about 130 ppm, where fresh litter samples, you don't see it at all. So this is just an example comparing litter to biochars, and you see that big peak centered at about 130 just is not there in fresh plant material at all. So if we could look for that peak, we could say how much charcoal is around. And so how we do it, there's actually two different experiments we could run on the NMR. One is the CP, or cross-polarization, versus direct polarization. And basically, the cross-polarization technique uses the extra energy of any hydrogen atoms centered around the carbon to basically see that carbon atom more clearly. And so obviously with charcoal, there's no hydrogen around it. They're just condensed carbon rings. And so it's basically underrepresented in the cross-polarization spectra. And so if we run it in the direct polarization mode, that peak should be relatively enhanced. And so this is the same example run in CP mode versus DP mode. And we see that large enhancement, and we could use that enhancement in that signal at that 130 ppm shift to tell us how much charcoal is present. The downside of this technique is the DP mode is extremely slow. We usually have to run a sample for at least 24 hours before we get the signal-to-noise ratio to an acceptable level where we could actually say something about it. So obviously we can't run too many samples in DP mode. We'd love to run everything, but it doesn't work. So what we're doing now is we're running a subset of samples and developing essentially ratios between the two. And there seems to be some pretty consistent trends between the two, and then basically applying that back to soils that we only run in CP mode. And so here's just a few more examples Jeff has put in. So we could see the soil on the top basically has no charcoal in it. We see almost no enrichment in that 130 ppm region in the DP mode. The soil in the middle has kind of an intermediate amount of charcoal, so there's a little bit of enrichment. And the soil on the bottom has probably 30-40% charcoal in it, if not more. And we see a really large enrichment in that 130 ppm peak. So if we get a range of soils with a range of charcoal contents and find what these ratios are between them, we could basically back apply those ratios to samples we can only run in CP mode because it only takes a couple hours. We can run them much more and get a much better picture across a wide range of soils what this charcoal content is. And then ultimately, we're doing all of that and trying to link it up with mid-infrared spectroscopy, which is really quick and gives you a lot of information about the soil, but it's not super quantitative, so we have to actually physically measure everything and relate it back to the MIR spectra. And so, for example, in the SCARP program, what we're doing, the program all up will have analyzed about 16,000 samples. So 16,000 samples will be analyzed for total carbon content and mid-infrared spectra, but we're hoping to get somewhere close to 10% of those samples through the fractionation process. So maybe 1,600 at most will go through the fractionation process, and then maybe only half that again, not even, will go through the NMR just because of the time constraints in a couple-year program. And so we could take that smaller bit of data as our calibration set and hopefully apply it to the mid-infrared spectra. And so this is just running through the process, and Les could probably answer any questions on this much better than I can since he helped develop most of this work, where basically we scan a bunch of soils, we get a bunch of spectra, we also analyze various data, so here it's probably just carbon content, and we use a partial least squares regression analysis technique to find what are the individual peaks in that spectra that most contribute to explaining the analytical data. So we could do this for any soil property we could think of pretty much. Clay content, iron content, pH, carbonate content, our different soil fractions, which we're most interested in here. So we create these loading spectra, and then we could basically predict what the carbon content or the carbon fraction content is of other soils that we haven't don't have the analytical data on. And we see that we do get, I think the next slide, so this will actually show you some measured versus predicted data. So this is from Liz's 2007 paper, where this is a set of samples from kind of around agricultural Australia. I forget the exact regions in particular, but you see we do a pretty good job, do a really good job predicting total organic carbon, slightly more variability in the particulate and charcoal pools. But it's not bad as something that takes a few seconds essentially per sample to get data on. So I guess the power in it is that we could easily measure lots of samples cheaply and quickly. And the downside is we accept that we're not getting perfect measurements because it's only a prediction. The prediction is only as good as the calibration procedure, and the calibration procedure never explains 100% of the variability, but it seems to work pretty well. And I think with the current programs, we have so many samples going through, we're actually going to be able to hopefully derive regionally specific calibrations, which are going to do a much better job predicting these carbon properties in different regions around Australia. So yes, linking, kind of talked about this earlier in the slides, this idea that we could link these measurable fractions into a modeling framework where most carbon models essentially conceptualize soil organic matter as more or less three pools: a rapidly cycling, an intermediate cycling, and a slow cycling pool. So this idea, we already talked about it, that we can link essentially the particulate fraction as this more rapidly cycling pool that turns over on the order of a few years, residence time of a few years in the soil, or humus pool maybe sticks around for a couple decades, so slower decay rates, and then the resistant pool essentially doesn't cycle on these management relevant timescales. So yeah, basically says that. I think this is just about it. Yeah, so the idea that, and this is essentially, so this is one of the calibration sites that I kind of mentioned earlier when I was talking about erosion work. It does do a really good job matching. So our model, our measured fractions, we could essentially model the measurable now, which is a really nice improvement over just modeling the conceptual. And this is work, there's research groups all around the world coming up with different fractionation schemes to match to model output, so we could get much more confidence that our models are, in fact, doing the right things for the right reasons. The models could be doing the right things for the wrong reasons. And if we could just match the trends in total carbon pretty easily, but if our conceptual basis for the model was wrong, we probably wouldn't be able to match the trends in the fractions as well. So it gives us much more confidence that the models actually are doing a halfway decent job in giving us some of the answer. So yes, there we go, last slide, summary. So fractionation, we can separate more biologically significant components of the organic carbon in the soil, provides a better understanding of management effects and subsequent vulnerability to change. This mid-infrared, partial least squares calibration prediction method can provide this nice cost-effective measurement capability on a large number of samples. And this idea that we could parameterize models with these measurable fractions to give us much more confidence in the model outputs. So yes, that's it. Thanks. Bill.
Yes, you've explained the changes in organic matter or organic carbon by the fractions. Now, there's some stuff that come out quite recently, particularly from Lehman's group, which suggests that the important thing is whether or not the organic matter is protected by clays and minerals. And they show that the protection occurs regardless of the nature of the organic matter, so that freshly inputted organic matter and also humified organic matter can equally be protected. Have you got comments on that particular approach?
Yeah, I mean, I guess, obviously I focused most of the talk on the kind of fractionation method that we use, but I guess going back to essentially like my second or third slide, that fits more into the aggregate hierarchy model, where essentially any organic matter, as long as it's encapsulated by soil minerals, is going to have, some protection to decomposition. So I guess that's more in line. I haven't seen that I still find it hard to believe that a little fresh bit of a legume, even if it's surrounded by clay, is as resistant as some microbial byproduct that's stuck really strongly. Just fresh. It takes a while for residues to develop the charge characteristics that are going to cause the strong binding that will create conformational changes in the organic matter that basically will render them inaccessible to enzymes. That's one of the reasons organic matter bound to clays doesn't degrade easily, because enzymes can no longer latch onto it. So if it's just a bit of residue encapsulated, it's going to be protected, but that encapsulation is surely going to be broken up at some point by a new root growing through, disturbing it or something, and then that stuff is accessible. But I think in general, I would 100% agree. In fact, some work I did a number of years ago, we used 14C to look at the age of carbon inside and outside of aggregates, and then we also ran an incubation experiment after breaking up the aggregates. We found stuff that was 2,000 years old based on 14C degraded just as easily as residues that were 5 years old once you remove that protection. So yeah, there's definitely, I guess it's that there's a disconnect between age and resistance, and I think you're right, and that's something that our fractionation scheme probably doesn't completely incorporate. And I guess this idea that there's a specific mechanism there, although with anything around soil carbon, there's tons of ifs, ands, and buts associated with it, I guess.
It obviously takes time for the clay to get around the organic matter, so
exactly.
therefore the very fresh stuff is going to be the most vulnerable.
Right. Ron?
Yeah, John. Just, I guess, continuing on from that, your results from the Kikuyu pasture study, where the POC did have a less negative delta 32, but it didn't seem to come all the way to Kikuyu even after 12 years.
Yeah.
Is that evidence for what John's saying, that you've got protected, what you're calling POC, but it's at least 12 years old?
Yeah, I think you're right. And I mean, you've seen more of these NMR spectra than I have. POC isn't just fresh plant residues. There's bits of organic matter that are greater than 50 microns in size that are pretty resistant to degradation in soils. And in fact, actually in Western Australia, we're finding this pretty much the strangest stuff in that particulate fraction where, especially in a lot of the really hydrophobic soils, you actually get coatings around the sand particles. And so you get this really highly aliphatic, like lipid-like stuff existing in what we're calling the particulate fraction. But in reality, it doesn't, what we're calling a particulate fraction might not really just be particulate organic matter in cases like that. So I think you're right. Yes, obviously our conceptual model that the particulate pool turns over on about a 5-year basis. In 12 years, we probably should have gotten about 70% turnover. And I think if you do a mixing model on those numbers, you get about 35% turnover, I think. So yeah, so there's stuff that's more resistant that is that's ending up in that particulate pool, definitely.
In regard to your residual organic carbon, obviously that's a very important fraction in the soil. Have you looked at mechanisms on how you could increase that residual organic carbon in the soils? And when you're talking about charcoal, you have no mention of phytoliths. So where do you bring phytoliths into your scheme of things?
Yeah, no, that's a really good point. That's something that, well, no, I guess I answered the first part first. Biochar, I guess, is the main management option that could increase that resistant carbon fraction. And so obviously there's a lot of research going on about how much biochar we could apply to soils, what type of, besides just obviously we could manipulate that. It's a pretty straightforward way to manipulate that fraction, but then what are the effect feeding back onto kind of plant productivity, what's happening when you're adding large amounts of biochar. So that's a very active field of research here and globally as well. And I guess in terms of phytoliths, that's something that we haven't treated specifically, and I know some research groups are really keen on phytoliths, and certain systems, like sugarcane systems, for example, produce lots of phytoliths. And I mean, they would essentially show up in the particulate carbon fraction, most likely. I mean, some would probably go through the sieve, but a lot of them would be retained in that fraction, and you're right. They would be, as long as they still had that silica protection, they would be much more resistant to decomposition than what our conceptual particulate organic carbon is.
So the phytoliths effectively can also build up that residual organic carbon with selectivity of crops and that sort of thing. So biochar is obviously one mechanism, but that's
that's a good point. I guess that would take it would take a huge management shift to a system to a plant that produces more phytoliths to really, I guess in the back of my mind, I'm always thinking options that farmers might take up are probably not ones that are wholesale shifts in the management system. But I guess you could breed within if it think it's going to be you could probably find varieties that produce more phytoliths than other varieties of any specific crop. So I guess that's a way of doing it. But yeah, it's interesting. I don't really have a good feel for what percent of the carbon might be in phytoliths in any given soil, so I can't really help there. Bill?
John, I think you showed quite well that the particulate organic carbon responded much better to management change. If thinking about sort of going into a carbon trading arena where we might measure total carbon, because it doesn't give us a good indication of the risk associated with a change back to another practice that might lose that more reactive carbon pool. Are we in a better position to get those measures more quickly now?
Off the record or on the record? You got to wait 6 months for the answer. It's looking pretty. It's looking pretty. The MIR work is looking pretty good. We're still. I think getting the particulate predictions are pretty good. The charcoal, there's still, I think, a slightly more question around whether we're going to be able to get really robust predictions or not. But I think Jeff's pretty optimistic that it's going to work out. If Jeff's optimistic, I'll be optimistic that it's working pretty well. We're not the only ones doing MIR style predictions. A bunch of groups around the world are using MIR to predict all sorts of soil properties, and it's becoming a pretty robust technology. So I'm pretty hopeful that, and like any modeling method, there's going to be errors around it, and that's just going to be something that's important, is that we carry those errors through, and we don't just report numbers. We report numbers with variability around them, and I think some approach like that should hopefully be reasonable in the future. And there is one of the outcomes of the SCARP program for us is we're hoping to develop essentially a calibration set of soils that other labs could then use to train their MIR, PLS techniques. It's every MIR instrument produces a slightly different spectra for the same soil, but it's just a matter of then calibrating your specific instrument, and then hopefully this is a technology that we're hopeful that a lot of labs, this MIR technology is a lot of labs have it. It's not that expensive. It's really easy to run, so hopefully a lot of labs around the country could take it up.